Hello, everyone. How are you all?
Good, and you?
I'm fine.
Good, thank you.
Good. Okay. So, ready to start?
Yes, ma'am.
Let's do it.
Yeah, let's do it. Let me see what's the time. Okay, just give me one more minute.
Okay.
I think people are still joining. Okay, so do you see my screen, the PowerPoint?
Yes, ma'am.
Okay, good. Hmm. Okay. So I think I talked about bioremediation, right? Just
Yes, ma'am.
after this. I think I should
Right
start
there.
from this part. Okay.
Mhm.
Let me move the screen over here. Okay, so let's start. And my pen color. So Monday I told that there are three objectives for this chapter. One was what is microbiology, or who are the microbes. Number two, actually, history of microbiology, like notion, early notions, and ultimately how we discovered microbes. Number three, how we are going to classify the microbes, or why do we need to classify the microbes, okay? So today we are going to start from the second objective. So second objective was early notions, or we can say the history of microbiology. Let's say history. Okay. So today we are going to talk about history of microbiology. Okay. So last week we talked about mostly about who are the microbes and how they actually help us. Okay. So now what happened? Like how do we know that there are some kind of organisms who are actually microscopic and we cannot see them without the microscope? So how do we know, or from when actually we are aware of the microbes? Okay, so today we are going to start from there, that how do we know the existence of microbes, okay, or from when actually we came to know the existence of microbes. Just a second, somebody is joining. Okay, so basically the prehistoric people, okay, prehistoric people, they were using microbes, and they were living with the microbes. Okay, people are joining. So prehistoric people, they used to live with microbes, and they were using microbes. Okay, like they used to make wine, they used to make pickle with all the vegetable. They used to store their vegetable in the form of pickle, different types of food, fermented food. They used to make. Okay, not only that. We found out Otzi the Iceman. Okay, so Otzi the Iceman, basically we found out a mummy. It's almost more than 5,000, I think 300 years old. And we found out this mummy underneath the ice of Alps, okay, near Austria-Italian border. So that mummy, that body was well preserved because it was under the ice. Okay, scientists, they found out that that person, that Otzi the Iceman, that person, he was suffering with some kind of parasitic infection. Okay, not only that, he was treating that parasitic infection or that disease with some kind of fungus, and scientists found out that fungus inside his belongings, with his belongings actually. Now that fungus, usually this fungus has a laxative and antibiotic effects, properties. So that means that that person, that Otzi the Iceman, he was using this fungus to treat his parasitic infection. Okay, so they used to use plants or something, some kind of organisms to treat all these type of diseases. Okay, they didn't know that it's a bacterial infection or parasitic infection or viral infection, but they knew that this fungus will help them, okay, to treat the disease or to control the disease. So basically what I told, that they used to use so many different types of plants, animals to treat all these type of microbes, okay, microorganisms. But they didn't know that they are actually, they got the disease from the microbes. So basically, unknowingly, or maybe they discovered the existence of some type of organisms who are invisible actually. So basically, so the evidence, scientific evidence shows that the existence of microbes is actually from the prehistoric period of time. And remember, Monday I told that the first organism on earth was a single-celled bacteria, that's the cyanobacteria. So anyway, so all those things like making wine, making a pickle, making fermented food, or using plants to treat the diseases. Not only that, like Greek, they had concept about a bad air. Okay, so they tried to improve their sanitization procedure. Again, just a minute, somebody. So, those Greek, they had idea how they actually came to know that bad air is not good for their health. Okay, so sort of they had idea the good sanitation or how to improve the sanitation system. Romans, in this picture, you can see this toilet, you can see. It's called Cloaca Maxima. Okay, so previously, before this, the sanitary process, the sanitary system, they really did not have idea. They used to do it anywhere. Okay, and that was unhealthy, but finally they came to know that it should not be like that. So they started making this type of sanitary system to control the sewer system. Okay, so you can see over here it was not that much modern, but you can see that multiple people used to sit together and used to do all those steps. But at least they actually discovered, you can say, or they found out that it would be much better if all those animal excretory products remain in one spot. Okay, or maybe towards the end of the village, towards or at the one point of at one place inside the city. Okay, so why they built this kind of building, or why they thought like that? That time we did not have microscopes, so they did not have any idea about the microbes, but at least they had the concept of the existence of some type of organism who are responsible for all these diseases. Okay, so all these concepts were going on, and all these things like fermenting food, using plants as to treat the disease, all these things actually instigate the thinking of the scientists of that era. Like Hippocrates, one of the, you can say, he was a physician, great biologist and a physician. We actually called him father of Western medicine. He proposed the idea that all the disease has a natural cause. They are not from supernatural causes. Okay, so during that time when there was no microscope, no concepts of microbes, so that time thinking about a natural cause, okay, and it was big thing. Okay, and that idea actually helped us to be here today at the 20th century. Historian Thucydides observed that cyberbirds of the Athenian plague were subsequently immune to the infection. He noticed that once the people get the plague, they get the immunity. Today, now we all know, specifically for the COVID-19, we every time we are talking about, the people who didn't even study the biology, they are also talking about the antibody. Okay, so what is antibody? How do we get that? That's because of our immune system, right? So now we know all these things, but that time that historian Thucydides, he talked about that immunity. He actually gave us an idea about the so-called antibody, what we are talking about. Basically, now we know that the plague people, if once they get that disease, that plague, body has that memory cell, okay, inside our immune system, the white blood cells, and actually our B cell and T memory cell, they actually make that antibody. Now we know that, right? But it actually, that thought process started from that time, from Thucydides. Marcus Vero, he proposed that disease could be caused by certain minute creatures which cannot be seen by the eye. So they had imagination. They used their imagination. They used their thought process. Okay, that is the reason we are here today, working with all the microbes. We can see the virus. We can even see the bacteria. Okay, but all these early nations actually paved the pathway of microbiology, of today's microbiology actually. So few things basically what we are going to work on. So the first thing, it was notion, early notion, that actually help us to discover the microscope. Okay, all those thinking, all those scientific thoughts, all those concepts that actually helped scientists to make the microscope and to observe the microbes. So Robert Hooke in the 1664 reported that living things were composed of cells. So Robert Hooke was the first scientist who first noticed a cell, and the name cell was given by him. He actually made lots of telescope, just single cell telescope and microscope. Okay, so he was observing some kind of specimen, cork specimen. He was actually working on cork specimen. Okay, under his microscope, his own microscope that he built up. That microscope was, as I told, that was only single-celled, a single lens microscope. It was not that strong, but he found out some kind of very small room type of structure. The room, it looks like the room in the monastery where monk used to live. Okay, very, very small, a minute room. So he actually felt the similarities between that monk's room and that cork space, and that's how he actually named them as cells. Okay, so basically the word cells actually was given by Robert Hooke, and he was the first person to observe the cell. Before Robert Hooke, we did not have any idea about the cell. Like all the organisms, all the any living matter is made up of cell kind of stuff. So we really did not have any idea. Antony van Leeuwenhoek, he was actually a cloth merchant. Okay, he also made his own microscope, and that microscope was much more powerful than Robert Hooke's microscope. Okay, so Antony van Leeuwenhoek, he was observing, as I told, that he was a cloth merchant, and he was observing on a cloth. Okay, cloth specimen, and he found out that very minute organism, and they are actually moving. Okay, so so that was the first time anybody saw the movement of the microbes. Okay, and he actually gave the name as, and he described all those moving minute organism as animalcules. And that's the reason he first observed any living cells moving. So Robert Hooke, he discovered the cell first. He noticed the cell first, but it was not moving. Okay, he really did not have any idea about that. But the Antony van Leeuwenhoek, he was the first scientist who noticed a living cell is moving, the movement of a living cell. Okay, so that is why he is known as the father of bacteriology and protozoology. Then after Anthony van Leeuwenhoek, so basically you can understand that we started observing microbes, or the existence of the microbes actually once the microscope was built up. Okay, with the presence of microscope only we can see the microbes, and we can prove the existence of microbes only with the microscope. Okay, not before that. So that the birth of microscope, or the origin of microscope, was very important for the field of microbiology. So you can say that discovery of microscope actually brings, or the reason was the reason of the birth of the microbiology, because before microscope there was no microbes, no concept of microbes. We really did not have any idea about microbes, okay, or the world of microbes. So so microscope, origin of microscope actually describe the birth of the microbiology, a new field of science. So after Robert Hooke and Anthony van Leeuwenhoek, so many scientists came in the picture. Here we will talk about very few. Louis Pasteur, 1857, he talked about the germ theory. He was the first scientist who talked like, okay, so you have disease, means some kind of microbes is responsible for your disease. Previously, those prehistoric people, they used to believe in all kind of supernatural power. Okay, they used to think that something you did wrong, maybe you lied, or something you did wrong, that's the reason you are sick. They did not have idea that your disease actually came from a microbe. So Louis Pasteur was the first person who proposed, who talked about germ theory. He told that no, behind every the disease, one microbes is responsible. Okay. And he told that fermentation is caused by the microbes. Okay, pasteurization, like how nowadays you drink your milk, juice, the pasteurized milk, juice, everything, that process, that pasteurization was discovered by Louis Pasteur. Robert Koch, 1914, another scientist who actually described the process of the identification of microbes. Means what are the steps we have to follow to identify the microbes from a disease. Okay, we still use the same procedure that Koch, it's called Koch postulate. We still use that Koch postulate to identify the disease. Like when you are sick, you go to doctor. Okay, what they do, they take blood from your body. Okay, so why they take blood? Because that blood carry all those microbes, the microbes who are responsible for your disease. Okay, then what they do, they actually culture your blood. They culture your blood means they actually allow the microbes to grow, the microbes inside your blood to grow. Okay, and they find out, okay, so what are the different types of microbes are there. Okay, and from there they actually diagnose the disease. So these are the steps described by Robert Koch, and it's called Koch postulate. There are 4 postulates are there. We are going to study all those postulates in second chapter, but still we are using all those postulates. So now let me go back. So over here, so we started with notion. Okay, all the prehistoric idea, all the scientists from prehistoric era, whatever they thought, their thought process or their discoveries, their opinions, from there we started, and that actually ignite to work on the microbes, or all these early notions actually ignite um, um, what you said, to work to find out the microbes. Okay, and that's how ultimately we discovered the microscope. Then next, so microscopy was there, and now scientists started looking at the microbes. Once microscope is there, once they started looking on the microbes, they found out that tremendous, so many different types of microbes are there, so many different types of shapes. So basically, the diversity was huge. Okay, in every aspect, in shape, size, color, their behavior. Okay, so all the scientists, they were confused. Okay, so ultimately what happened, so there was necessity to basically organize all those, you can say, the systematically organize all those microbes. Okay, so after development of microscope, the diversity of microbes actually bring us to a situation where scientists realized that we really need to systematically organize all the microbes. Otherwise, we all are lost. Okay, because of there so many different types of shapes, size, color, and the behavior. So from there, when they started thinking about the systematic organization, okay, so that means a new branch of science came out, and that's called the taxonomy. When we started systematically organizing, so basically all these things are related to each other. One actually is the reason of the other. So now at this moment, we are on taxonomy, a new branch of science. Okay, so taxonomy, what it does, it's actually organize, it's classify, and it gives the name. Okay, our earliest scientist, most earliest actually, who actually worked on this taxonomy was Carolus Linnaeus. Okay, Linnaeus, he published a small book, and the name of the book was Systema Naturae. It's a published paper. Over there, he worked on three groups, three actually different topics, you can say. One is the classification, then the nomenclature, and the identification. So what he told and how he systematically organized all the organisms on Earth is called, that's a new branch of science, and it's called taxonomy. Okay, it's the lineal the taxonomy was most basic fundamental one. Okay, so he told, the first you have to identify. What you have to identify? You have to identify the characteristics, features of the organisms. Okay, so this organism, it has two limbs. Okay, one, so this has two limbs, maybe based on limbs. Okay, so till 2 limbs versus 4 limbs. Okay, so all the 2 limbs, the organisms who has 2 limbs, they will be in one group. All the organisms who has 4 limbs, they will be in separate group. So based on the number of limbs, he found out 2 groups. Exactly same way, the presence or absence of hair, he grouped all the organisms. So all these, like hair, presence of hair, presence of or the number of limbs. Okay, where do you live? Are you aquatic or terrestrial? So all these are visible characters. And based on all these characters, he first identified all those similarities between them. Okay. Then he gave the name. Okay, so different groups had different names. Not only that, under all the groups, under one group, all the organism was given name, and this assigning name is called nomenclature, and then classify. Okay, so basically, you have 4 limbs or 2 limbs. Okay, so so you are in this group, and all the organisms were given name. Now, based on all those similar characteristics, he actually classified all the organisms. How he actually classified? What he did, he basically, Linnaeus, what he did, he first divided all the organisms on Earth. He divided on 3 groups: animal, plant, and mineral. So previously, he actually thought that minerals are also living organisms. That's what he thought at the beginning. Okay, because lots of stones are there, they change their color and all those. So that's what he got confused that time. So he first divided all those organisms into 3 groups: animal, plant, and mineral. And then later he realized that minerals, they are not living organisms, so he took out mineral. Now all these organisms, so animal and plant, he divided all these under different levels and sub-levels, okay, based on the hierarchy. So first, uppermost level was kingdom, then class, then order, then family, then genus, and the species. Species was the smallest one and the most specific one. So mostly lineal classification was based on the outside morphology, by the visible characteristics. As I told, like how many limbs are there, presence or absence of hair, all those things. With the invention of technology, slowly refinement of linear taxonomy actually occurred by different scientists, and we started working on the phylogeny. Phylogeny means we started grouping based on the evolutionary relationship. Okay. And best way to do that was drawing a phylogenetic tree. You can see all this picture, these are the phylogenetic tree. Previously, this phylogenetic tree we used to, or the scientists used to make based on the similarities, like how many limbs are there. Okay, so all they have the limbs, so they can make one tree. So limbs, or they have limbs, so they have 2 limbs, they go over there. They have 4 limbs, they go over there. So that's how they started making the trees. But nowadays we also work on the genetic, biochemical, and embryological evidence also. At this moment, we only work on the genetic, mostly the genetic, and not only the DNA. We work on the rRNA. We work on the protein. Okay, but still we make that phylogenetic tree. Ernest Haeckel's, he actually, at the beginning, he with linear taxonomy, with animal and plant, he added one third kingdom, that's the Protista. Okay, so we started with linear taxonomy. We had two kingdom. Okay, that time we had two kingdom. Now Ernest Haeckel, at the beginning, he added one more kingdom, that was Protista. So plant, animal, and then come Protista. Okay, so at the beginning, now all these like plant, animal, and protists all are eukaryote. Okay, all are eukaryotic organisms, means they have specific nucleus, they have membrane bound organelles. Okay, they're developed organisms. Okay, and he realized that, and then towards the end of his life, basically almost before his death, he added the fourth kingdom, that was Monera. Monera means he actually included all the prokaryotic unicellular organism over here. Okay, so now you can understand that we are complete. We have all the eukaryotic organisms over here, and over here we have all the prokaryotic organisms. So lineal taxonomy, then Haeckel's, at the beginning Monera was not there, so animal, plants, protist, and then he added Monera. Then Robert Whittaker, he added another kingdom, and that was the fungi. Fungi, he discovered that fungi, they looks like plant, but they are not plant. Okay, so plant-like organisms who are actually not plant, they got included in the classification. And now we all know that plant, they have cellulose, okay, but the fungi, they have chitin. So that fungi, they looks like plants, but they are not plant because they do not have cellulose, they have chitin cell wall. Then our latest phylogenetic tree came from Carl Woese. He was a geneticist. He actually, based on the ribosomal RNA, he drew this phylogenetic tree. And what he did, he actually gave us domain. So above the kingdom level, he actually made one more level, and that's the domain. And based on Carl Woese, there are three domains: bacteria, archaea, and eukarya. Okay, so bacteria and eukarya both are actually prokaryotes. Okay, so sorry, bacteria and archaea, they are both are actually prokaryotes. Okay, so they are very, very similar. They are single cell unicellular organism, and both are prokaryotes. But if you see the phylogenetic tree, you can see that we all originated from a common ancestor. Okay, probably that cyanobacteria, like archaebacteria, you called those as archaebacteria. We all originated from a common ancestor. Okay, and that heritage actually went for a long time. Then two groups, it's divided into two groups. One went this side, one went that side. So from this side, we got archaea and the eukarya. And this side, we got all the bacteria. So before Carl Woese, okay, if we compare the Whitaker or lineal taxonomy, you can see that if it is morphology based or embryological based, then we could see that bacteria and archaea, they are very closely related. But from the rRNA classification, it came out that archaea are much more closer with us, with the eukaryotes, than the bacteria. Okay, you can see that we all are, both archaea and eukaryotes originated from one ancestor, one common ancestor from here. Okay, and then they divided, they get separated from each other. So that's our latest classification, and we still follow Carl Wu's classification. So now at this moment, this is the levels of classification, and we start with our uppermost one is the domain, then kingdom, then phylum, then class, order, family, genus, and species. Okay, so make sure you memorize, serially. So first kingdom, then phylum, then class, then order, then family, then genus, and species is the smallest one and very specific one. So that's the classification. We up to this, we talked about only one part of the, so basically we have done the two part. Like over here, I told that first the identification. Identification means the similar characteristics, okay, similar features. So that's the identification. Then classification, okay, based on the similarities or dissimilarities, we started classifying them, and we are over here now at the nomenclature. Now we have to give the name. So what happened? Why we have to give the name? Because let's say some people, like somebody is coming from India, okay, and he has some type of disease. So over there in India, people, a doctor said that it has the name of the bacteria is called Neem. Okay, over here in US, we really do not know any bacteria is called Neem. Okay, so if we all are not on same plate internationally, then it is very difficult to diagnose or to identify the bacteria or identify the microbes. Okay, so we actually developed a special system of naming, and it is called binomial nomenclature. Binomial, the word means it has two names. Every organism has a two-name designation, a standardized nomenclature that allows scientists from all over the world to exchange the information. Okay, so like over here in India, if doctor says that it is Escherichia coli, then over here US we know, okay, so it will not take time. I hope you remember that few years ago that Ebola came, right? Okay, so Ebola virus came from the Africa, South Africa. They have whatever the time they got to work on the virus, they gave their own name, okay, in their own language. People from South Africa or the scientists from South Africa who had here, they came. We really did not have idea. Even we can talk about the coronavirus. We really did not. Still, coronavirus. China actually told that it's a kind of SARS virus. At least we had a little bit of idea. But during the case of Ebola, we really did not have any idea that African people, African doctors and scientists, they gave us name. Okay, but that's in their own language. And we really did not have idea. It took so much time for us to identify that virus. It took time, and by that time to know the virus, to identify the virus, to know all the characteristics, features, how it reproduce everything, by the time we came to know, it was too late and we got the infection. Okay, so I hope you understand the importance of giving the scientific name so that we all are in the same plate, on the same plate throughout the world. Otherwise, it will happen. Okay, somebody will bring the infection from some other country, and over here we will take a long time to identify virus or bacteria, and it will infect, then we will be in trouble, just like coronavirus. We are suffering. China told that it's a SARS virus. Still, we are, it is still taking us time. So that binomial nomenclature. So what is binomial nomenclature? So two parts should be there in each name. Like I told you, E. coli. Okay, so basically the full, let's say over here I gave you example, Staphylococcus aureus. So first name should be generic name from the genus, and the second should be the species. So Staphylococcus, the generic name is the Staphylococcus, aureus is the specific name. Now, how do we write down? So generic name should be, the first letter of the generic name should be capitalized. You can see that Staphylococcus S is capitalized. Species name starts with lowercase. Okay, both the name, generic and specific name, should be italicized or underlined. This slide is very, very important for the lab. Lab, your skill test, you have to identify one unknown bacteria. Okay, and on your lab, basically you have to write down the scientific name. You have to identify which bacteria I gave you. If you do not write down the scientific name properly, you are going to miss all 40 points. That identification itself, that scientific name, writing the scientific name properly carry 40 points. If you cannot write it down, if you do not know how to write down the scientific name, then you are not going to get that 40 points. And I am very, very strict about it. Okay, so make sure you really need to know how to write down the scientific name. Let's say Bacillus subtilis, one bacteria. So Bacillus is the generic name, and subtilis is the specific name. Okay, so we know that generic names should be, the first letter should be capitalized. So Bacillus, B should be uppercase. So Bacillus, and specific name subtilis. We will start with lowercase. So small s, subtilis. So generic, capitalized, lowercase. Now the next rule, either italicized or underlined. If you type it, then it has to be, all these names should be italicized. But if you are writing, that means you have to underline. And underline, two separate underline, one for generic name and one for species name. There should be gap in between. Sometimes students, what they do, they just make one underline. I give 0 for that. Okay, so just these are the 3 rules. Make sure you know. Every time you can see, like E. coli, Escherichia coli. So E should be uppercase, C should be lowercase, and every time I used separate underlining. Do you have any question till now? Anybody?
Are we going to learn how to get the species name, or are you going to provide that for the
That I will write it down on board in lab.
Okay.
So there are, I think, 6 or 8 specimens. Out of 8, you have to identify one. And all those 8 specimens, you know beforehand that these are the 8 specimens, and out of 8 you are going to get one unknown. But make sure you know the spelling, and you really need to know how to write down the scientific name.
Okay.
Okay.
So you were saying that we need to double underline when we write it?
Not double, just one.
Oh,
One
okay.
underline, but it should be separate.
Okay.
Like this, like just one underline here, one underline here, but it should not be like this. It should
Okay.
not be continuous. It should be separate underline. One
Okay.
for generic name and one for species name.
Thank you.
Okay? Anybody? No question? Okay. So for bacteria, we use Burgess manual. Okay, Burgess determinative bacteriology. So basically, Burgess manual, it is called determinative bacteriology. Still, we use this book. It's a huge manual. I think 12 or 14th segments are there, and all the characteristics, basically all the information to identify bacteria you can find inside the Burgess manual. I do have that Burgess manual, determinative bacteriology, and one day I will take it to lab to show you how actually we classify the bacteria. So types of, basically in this slides, we talked about the size of the microbes. They are really, really small. The relative sizes of various microscopic and non-microscopic objects. Note that a typical virus measures about 100 nanometer, 10 times smaller than a typical bacterium, which is like 1 micrometer. So you can see one atom, 0.1 nanometer, okay? And one poliovirus is almost 20 nanometer. One bacteria, that's the average size we are talking about, in general, 1 micrometer. So you can understand, but the red blood cells is much bigger than the bacteria. Okay, so you can understand the actual size of the bacteria. They're very, 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 very small, very minor. So all the microbes, last Monday I talked about 2 different types of cells, prokaryotes and eukaryote, and we talked about that who are the prokaryotic microbes and who are the eukaryotic microbes. So we will go a little detail. So prokaryotic microbes, who are prokaryotes? Bacteria and archaea. Bacteria, they have specific shape. Usually, mostly 3 different shapes are there. One is round, and we called those as coccus. So cocci bacteria. Rod-shaped bacteria, they are called bacilli. Okay, and the spiral-shaped bacteria called spidillum or spirochaete. So these are the 3, shape of bacteria. So coccus, bacillus, and spirochaete or spidillum, spiral-shaped. Round-shaped bacteria, they are called coccus. Rod-shaped bacteria, they are called bacillus and spidillum. Now, sometimes what happened, few bacteria, so these are the coccus, bacillus, and spidillum. These are the 3 most common and general term. sometimes they're very, very rare, very few bacteria are there that they are actually rod-shaped, but there is bend, okay? It's carved. So carved rod-shaped bacteria are called Vibrio. Now the Coccobacillus, you can see they are not really coccus, not really round shaped, not really bacillus, okay? But you can say mostly maybe rod shaped or circular. So they are called Coccobacillus. So these are the few general shape of bacteria. We actually classify bacteria based on their shape and their arrangement. Okay, so we will work on the arrangement of bacteria in lab, but these are the regular shape. Archaea, they actually look like bacteria, single cell organism, okay? And prokaryotic cell, they do not have nucleus, no membrane bound organelles. But one very interesting characteristics about archaea is they are called extremophiles, means they are extreme lover. So anything extreme, they love it. Means high acid region, high acid, they can tolerate. High heat, they can tolerate. High salt, they can tolerate. Okay. Only difference between bacteria and archaea is the cell wall structure, and we will work on that I think in chapter 3. So there is little difference in cell wall structure between archaea bacteria, but otherwise, and only the behavior, otherwise they look alike, they behave very similarly. Now eukaryotic, under eukaryotes, there are 3 groups. One is protist, then the second is the fungi, and helminths. Protist, both unicellular and multicellular. Okay, they are algae and the protozoa. Algae, they are plant-like protists, but they are not plant. They are photosynthetic like plant. They have cellulose cell wall like plant. We used them for the ice cream, to make salad dressing, lipstick, toothpaste, biofuels, all those. They're very, very helpful. Okay. So, but there are very few algae who actually are responsible for human diseases, very few. And for this course, we really are not going to study about algae in detail. We will study very little bit, but we will not going into detail. Protozoa, there are lots of different protozoa. There protozoa, you will see that it's a huge group of organisms and who are not under any specific taxonomical level, okay? Because scientists are still working on this group, this protist. You can see that algae, multicellular large organism, okay? You can see with your naked eye. Protozoa, some of the protozoa, they are most of the protozoa actually they are microscopic. There are few you can see without a microscope. But the protozoa, very important characteristics features, they have locomotory organ, either cilia or flagella or pseudopod, so that they can move. Okay, that's the characteristic features of the protozoa. So you can understand that then over here, maybe it's a amoeba, one single-celled organism versus algae, large, you can see the green algae, okay, inside the ocean, underneath the ocean floor, it's huge. But this single cell protozoa and multicellular algae, they are under same group, and that group is called protist. Because the scientists, there are lots of organisms we couldn't finish classifying, or we couldn't trace the classification, their origin. So basically we are not done working on those organisms. What scientists did, all the organisms who are not properly identified, they actually kept all those organisms under protist. So protist, it's a huge group of organisms and very little similarities between all those organisms. So anyway, protist, for microbiology purposes, we are only interested about algae and protozoa. Okay, but for this course, we are not going to cover algae, but we will work on the protozoa. Then the second, the fungi, plant-like organism, but they have chitin cell wall. Two different types, both unicellular and multicellular. So over here, protists, they are both unicellular, multicellular. Fungi, unicellular, multicellular. Unicellular fungi, example is yeast. Multicellular is mold. Mold, it's not microscopic. You can see it with your naked eye. Helminths, multicellular parasitic worms like Platyhelminthes, Nemathelminthes. Adult organisms, they are not actually microscopic. I talked about it, but eggs and larvae are responsible for the disease, for our disease, for human disease. So that, and eggs and larvae are microscopic. That's why they are included in this course. So these are the three groups of eukaryotic microbes. Now the last group, acellular. Why acellular? Because they do not have cell, proper cell, not independently living. Okay, much simpler than cells. Basically, a small amount of DNA or RNA wrapped in protein and sometimes by lipid membrane. So they really do not have cell. Remember last Monday I talked about what are the characteristic features of a living cell. They can do metabolism, they can reproduce, they can grow in size. Okay, they must fill the outside because basically they must have sensitivity, but virus, they really do not have any of those characteristics. They cannot do metabolism, they cannot reproduce, they cannot multiply by themselves. They are not even living outside of any living cell. To replicate or to multiply, they must have a living cell. Okay, like COVID, we are now at this moment, we all are, so scared about that coronavirus. It's a virus. So it has RNA, it's an RNA virus. Okay. The interesting features of a virus is they don't even have both DNA and RNA. So inside our cell, inside a single cell, we have DNA and we have RNA. That these RNA, they help us to make our protein. They help in protein synthesis. Okay, most of these RNAs. Virus, they do not have both DNA or RNA, DNA and RNA, either DNA or RNA, and that's how we actually give them name. Like it's a DNA virus or RNA virus. Like coronavirus, it's an RNA virus because it does not have any DNA. It has RNA. Okay, so virus, typical virus, they have either DNA or RNA. Then outside they have a protein layer. Okay, very thin layer of protein. That actually a protective layer that protects the DNA or RNA inside. That's their genetic material. If you can say that it's a genetic material, yes, that's the genetic material, and it is protected by that protein layer, and that protein layer is called capsid. There are a few virus who has envelope outside of that protein. Okay. Interestingly, these envelope is actually made up from host cell envelope. Like if this virus, like let's say if it is coronavirus, okay? So coronavirus, it's mostly human virus, right? Animal virus. So like if it is inside your cell, your plasma membrane, your cell's plasma membrane will be very similar with this virus's plasma membrane, because this envelope is made up from your cell membrane. Okay, so basically all the carbohydrate thing, the sugar, polymer, they grab from the host's membrane. Only the genetic material is their own. The protein is it's a viral protein, but they force the host cell to make that protein. Okay. Individual virus, mature individual virus is called virion. Scope of microbiology. At this moment, you can understand that we have trillions of different types of microbes and so many trillions of different types of diseases. Okay, it's a huge field of biology. So there are so many different area of microbiology, like bacteriology, who study only about the prokaryotes; mycology, who only study the fungi; phycology, who study the algae; protozoology, the scientists who study about the protozoa. The study of viruses is called virology. Immunology actually include all these organisms, okay, like bacteria, fungi, algae, protozoa, viruses, and they work with the immune system. Okay, basically how immune system behave with all these organisms, that's the immunology. So these are the different scope of microbiology. This slide is also very important. Make sure to memorize all the definition from this slide. Okay, I usually give at least one question from here. Oh, okay. So we are, chapter 1 is done. Do you have any question from chapter 1? Any question? Did you get it? Did you understand that what it is about?
Yes.
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 So just remember again the summary. Just remember three things. That objective is like, first we are actually going to work on who are the microbes, okay, and what is microbiology, why should we study about them, all those things, okay. Then from when actually we came to know, how we came to that point, how we discovered the microscope, or what actually inspired to discover microscope. Okay, what are the reasons behind the making of microscope? So all those things are there. Then the taxonomy, how actually microscope helped to make a new field of science, the taxonomy. Or you can say the new field of science, biology, the microbiology. How we came from there, from the microscope to taxonomy, microscope to microbiology. Okay, so the development of microscope actually, you know, that's the starting point of microbiology, and that's the starting point of taxonomy, you can say. So if you understand all these link, then this chapter is very easy. Few definitions are there, bioremediation, all those things are there. The scientists who did what, you have to memorize that. Otherwise, this chapter is very easy to go. What do you say?
So then the last part where you said the 3 questions, what is microbiology, history of microbiology, and then the third one, how do we classify the microbes? That would just be like the tree, things like
Yeah,
that.
the classify, like the first question, the classification, the taxonomy. Why do we need to classify them? Why? Because the diversity. So many different types of microbes are there. The shapes, the size, their behavior. You can understand that the algae, it's a completely plant-like. We make ice cream from that. Okay? And one protozoa, amoeba, they can go inside your brain and they can eat your brain-eating amoeba. So you can understand the diversity, enormous diversity. So they are so different. If we cannot classify them, if we cannot group them, then it would be very difficult to study. And that's why that taxonomy came. That's why that classification came. And classification, that's the reason of classification. And from there, the microbiology starts. The new field of biology. Before that, it was not there. There was no microbiology before microscope.
Wonderful, thank you.
So do you have any question? No? Okay, so today is Wednesday. Coming Monday, I will start, chapter 2. Okay, I'm not going to start today. Today, weather is so bad, I'm losing internet connection. I hope you all are safe and doing good. So our lab, from next week onwards, our lab is going to start. Few things I want to tell. Look, if you have, before you come to the school, come to the building, make sure to take your temperature. Okay. So make sure to take your temperature. Then if you think that you are sick, you are not feeling well, you have headache, you have coughing, breathing problem, please do not come to the class. We have instruction that anybody is coughing inside. If you cough one time, I have to shut down the lab immediately. Okay, that's the instruction we are given. So basically, if you are not feeling good, please do not come in the lab. Text me or email me. I will help you for that lab. Don't worry. If you are sick, if you cannot come, I will help you to get all the material for that lab. Plus, but please do not come to that building. That's the one thing. Number 2, please bring your mask. We are going to maintain social distancing, so I will not go to you. Okay. Probably I will have microphone, so I will be loud enough. And I will post the announcement on your D12. Make sure to check the D12 every day, because all the information, that's the only source to get all the information. So, and if you are not done with your syllabus quiz, please finish that syllabus quiz. It's very easy, 5 questions are there. Read the syllabus and finish it up. Okay? If you have any question, any concern, email me.
What time do we meet for the labs?
That you have to see that it's in your detail, because this class is for all four section of labs. All the people from morning, Monday, Wednesday morning lab, Monday, Wednesday afternoon lab, Tuesday, Thursday afternoon lab, and Tuesday, Thursday night lab. Everybody is here. Because of the social distancing, like I am overloaded. So I really do not have time to go for separate lecture or separate office hours. So this is the time all four groups are coming. So your lab time, you can find it inside your syllabus. The front page of your syllabus will say the lab time of yours. I will post another announcement before the lab in detail, describing your time and rules and regulations and everything. Please continue to check the detail.
Okay, thank you.
Anything else? Any more questions? No? Okay, so I am then ending this meeting.
I have one, but it doesn't really apply to the
Yeah.
whole group.
Yeah, tell me.
So I know when they emailed you about getting married on the 28th.
Oh, you are the person.
Yes, ma'am.
Oh my God. And you are Mike, right?
Say that
James.
again.
What's your name? I can't.
James Craft.
James. Okay, so James, congratulations. First of all, congratulations. I mixed up with Mike. Okay, so Monday, I congratulate Mike. I thought, and he was upset, pretty upset.
I saw that. I was watching the lecture video earlier.
Okay. Sorry. Yeah, tell me.
So the 31st, I'll still be in Wisconsin because that's where we're getting married. That's where my wife's from.
Uh-huh.
And I was wondering, instead of coming to the 31st lab, could I just come to the 24th lab and then resume my regular lab schedule after that?
I don't think so. I have to see the chart. I really do not have the chart at this moment in front of me. I'm
in the A group. I know you're trying to keep the students to 8 or 9 or less. So the A group is 8 people, and my group's 9. So I wouldn't imagine it'd make a whole lot of difference because I mean it would still be under the required 10.
So you are not coming. You will be absent both the days, right? Monday and Wednesday.
No, I can make the Wednesday lab on what is that? The September 2nd. I can make that lab. I just want to know if I could like swap my Mondays around so I can still attend.
What time is your lab?
It's the Monday, Wednesday, 9:15 to 10:45.
Okay, so your first lab is 24. So 26th you will be here.
So I'm in the B group technically.
B group. Okay,
so I've got the 31st and the 2nd. I was wondering if I could swap the 31st with the 24th.
No. Okay, that's fine. So 2nd September you will be here, right?
Yes, absolutely.
Okay, then you should be fine because 31st, the first day of lab, we will just work on the syllabus, rules and regulations and all those. And then after that, if we have time, I will start working with the microscope, okay? And we will still continue working on microscope on Wednesday. So you are not actually going to miss anything.
Okay, so you
make sure to.
I'm just wait until the 2nd.
Yeah, so don't worry about the 31st. You should be fine. Only thing you have to download one form, okay, and you have to sign. That you can do. You can bring that form on 2nd.
Okay, it sounds awesome. Thank you.
Yeah. So basically, for the social distancing, I am really restricted. Otherwise, I wouldn't have any problem. Anybody come come and join it there if there is any problem. But we are so restricted. We should not allow more than 8 people. And I am also a little bit scared and all those. So, so that's the problem. But I see that if you are not missing Wednesday's lab, then you should be perfectly all right. You should be fine.
Okay, thank you.
Okay. And if you need a little bit extra time, that's also fine that day. Okay.
Okay,
Okay. Congratulations again. Enjoy.
thank you.
Anybody? No questions?
I have a question.
Yeah. Tell me.
So the first lab, is it going to be is it next week or
Yeah.
the 1st of September? Like in-person lab?
Yeah. In-person lab. It is going to start from Monday. Which day you have lab? Monday, Wednesday?
Monday the 24th, yeah. 24th,
okay.
Monday.
Yeah.
So, okay, so we'll be in school Monday the 24th, right?
Yeah.
Okay, thank you.
Okay.
I had
Any
a
more questions?
question.
Yeah. Tell me.
Is there any way that you could post any of the due dates for the material, like the discussion questions, or
I already posted.
they are?
Uh-huh.
I didn't see the due dates.
No,
on
go to content. Then you can see that discussion assignments.
Right, but the due dates for them, like when they're
Inside just go over all this content. If you so just click the discussion 1 content. On the top you can see the due dates. It's there.
okay.
Go to content, then discussion assignments, then just click discussion 1.
Oh, my computer just crashed.
And it should show in your calendar also.
Right, I checked in the calendar and I didn't see anything, or I'm just looking in the wrong spot. But
I checked, but it should be there that inside the calendar. Because when I set it up on that way that everything should show in your calendar.
okay, for some reason my computer's all frozen on D2L.
Okay, I will post all the
I'll
dates.
email you.
Don't worry, I will post. Okay, let me write it down, and I need to post discussion dates, right? I will do that. And if you go to the assignment folder, up assessment, then go assignments.
Right.
Yeah, and over there you can see all the dates also.
Okay.
But
So the first one isn't due until September 30th.
September 30th, because there are a few chapters you really need to read, you really need to study. Before
Okay.
that, because all these assignments I gave from the concept that we are going to study, to learn. So you really need to read few chapters, then only I can expect your opinion, not before that. So first September 30th, and then every 15 days you have one assignment due. So I think September 30th, then the next assignment is October 15th, then the third one should be October 30th, then November 15th, and then December 1st. I think every 15 days you have one assignment due.
Alrighty.
So the
Thank
first
you so
one.
much.
Anybody? Anybody else? No question? Okay, so good night then, everyone. I will see you on the lab Monday, Wednesday, and next Monday during this time. I will go over chapter 2. Okay?
Okay, thank you. Good night.
Okay, good night.
Thank you.
Bye.
Thank you. Bye.